In this lecture, we will talk about oxygen transport and hemoglobin. We will review the structure of hemoglobin and its role in gas transport. We will use the oxygen-hemoglobin dissociation curve to explain the transport of oxygen in the blood. And we will describe the Bohr and Haldane effects on hemoglobin saturation. So we've already talked about gas exchange between the alveoli and the blood. We've also talked about gas exchange between the blood and the tissues. In order for that exchange to take place, we need to transport oxygen and carbon dioxide in the blood between the lungs and the tissues. There's two main ways for oxygen and carbon dioxide to be transported. They can either be dissolved in the plasma or they can be bound to hemoglobin. For oxygen, the majority of oxygen is carried on hemoglobin. Hemoglobin makes up 97% of the content of the red blood cells. It has a 4 subunit structure with 4 heme groups containing iron and 4 globin polypeptide chains. In adults, that's 2 alpha globins and 2 beta globins in general. Oxygen molecules will bind to the iron within the heme group, giving each hemoglobin the maximum carrying capacity of 4 oxygen molecules. And we can see that drawn here, where we have an oxygen connecting to each heme group. So that's 1, 2, 3, 4 possible oxygens on one molecule of hemoglobin. It's not only important for the hemoglobin to bind the oxygen, but also to release it when it reaches target cells. So hemoglobin changes its structure for binding and unbinding. When hemoglobin is bound to oxygen, it's oxyhemoglobin, and it takes on a red color. When hemoglobin is not bound to oxygen, it's deoxyhemoglobin, and it turns more of a dark red or bluish color. Hemoglobin can also bind carbon dioxide. It's not the primary way in which carbon dioxide is carried, but it can bind to carbon dioxide, and we'll talk about that importance when we talk about the Bohr and Haldane effects. Carbon dioxide doesn't bind to the iron groups or the heme groups. It binds to the globin chains at a separate site on the hemoglobin. When carbon dioxide binds to hemoglobin, it's called carbaminohemoglobin. And again, the majority of carbon dioxide is not carried on hemoglobin, it's carried within the plasma. We'll talk about that in the next lecture. So for this lecture, let's focus on oxygen. Oxygen is mainly transported bound to hemoglobin. It does not dissolve well in plasma, so only about 1.5% of the total oxygen in the blood is carried dissolved in the plasma. The other 98.5% is carried on hemoglobin. So hemoglobin is specialized to transport oxygen, and we have, as we said in the previous slide, up to 4 oxygen molecules per hemoglobin. This is the reaction that shows hemoglobin binding, but also unbinding to oxygen. As a clinical note, hemoglobin levels will increase in individuals that have impaired gas exchange. That's an attempt to get more oxygen carrying capacity in the blood. So hemoglobin needs to bind oxygen in order to carry it from the lungs to the systemic circuit, but it also needs to unbind it in order to deliver it from the tissues. We call this the loading and unloading of oxygen from hemoglobin. This happens based on the oxygen gradients and the other molecules surrounding hemoglobin. Hemoglobin will change shape in a high oxygen environment to bind more oxygen. And it will change shape in a low oxygen environment to release and give away more oxygen. I think it's like Robin Hood. We steal from the rich and give to the poor. So oxygen should bind in oxygen-rich environments, and oxygen should release from the hemoglobin in oxygen-poor environments. So this is the loading and unloading of oxygen as hemoglobin changes its shape. This is based on the partial pressures of oxygen. So the partial pressure is essentially a measure of the percentage of gas in a given mixture. In this case, we're talking about the percentage of oxygen within either the alveoli or the pulmonary capillaries or the other areas of gas exchange. Partial pressures will determine the hemoglobin loading versus unloading at each site. When the partial pressures of oxygen are high, the oxygen should bind and load onto the hemoglobin. When the partial pressures of oxygen are low, the oxygen should unbind and unload from the hemoglobin. If we think back to our gas exchange lecture, we want the loading to occur at the lungs, where we're oxygenating the blood, and the unloading to occur at the tissues, where we're dropping off the oxygen for cellular respiration to take place. As hemoglobin loads and unloads, the sites of hemoglobin binding can be visualized in this model of hemoglobin where we have 4 available sites. We can see that as those sites fill up, the hemoglobin will become more and more saturated with oxygen. So hemoglobin saturation refers to the amount of oxygen that is being carried on each hemoglobin. This is represented on a hemoglobin saturation curve, where hemoglobin is more saturated as the partial pressure of oxygen increases, and unloaded or less saturated as the partial pressure of oxygen decreases. I want you to be able to picture this saturation. So let's think about what these percentages will look like. Remember, we have 4 possible sites on each hemoglobin. So if we look overall at a sample of blood, if the majority of hemoglobin has all 4 sites bound, then we say that that is nearing 100% saturation for hemoglobin. If we have no oxygen bound on average in that sample of blood, then we have a 0% saturation. And then we change by 25%. 1 out of 4 possible sites on average would be a 25% saturation. 2 out of 4 possible sites on average would be a 50% oxygen saturation. 3 out of 4, a 75%. So keep this saturation in mind, and now we're going to graph and look at the change in saturation based on the amount of oxygen in the area near the hemoglobin. So the hemoglobin saturation curve is the way to visualize this. I'm going to draw one for you, and then I'll show you the textbook one. On the y-axis of the hemoglobin saturation curve is the percent saturation of hemoglobin. As we just said, we can go anywhere from 0 to 100% saturation. So 0 being 0 out of 4 sites bound, and 100% being 4 out of 4 sites bound. So we're going to range from 100% saturation to 0% saturation. And then we can mark in between. 25, 50, 75, 100% saturation possible. Now we want to ask, how does the hemoglobin molecule respond in saturation based on the partial pressure of oxygen that it's in contact with? Think back to our gas exchange lecture. What is our range for partial pressures of oxygen? Our range is typically between 100 and 40. So our max is going to be somewhere around 100 millimeters of mercury. 40 is approximately what we would see at the tissues. And of course, just to keep it even, we can go all the way down to 0. So on the x-axis, we have partial pressures of oxygen. And now we want to say, what does hemoglobin do? How does it respond in an oxygen-rich versus an oxygen-poor environment? And as we said, when there's a very high amount of oxygen in the environment, the majority of hemoglobin will be bound, meaning we'll have 4 out of 4. So here at 100 millimeters of mercury partial pressure of oxygen, we'll have 100% on average saturation of our hemoglobin. And that holds pretty stable for most ranges of partial pressures of oxygen. It's not until we start getting down into the very low ranges that we have a quick drop off in the saturation of hemoglobin. So now we're going to see it drop off fairly quickly. So this is where the hemoglobin saturation curve forms an S shape. And what that means is that when the hemoglobin gets into very oxygen poor environments, it's going to see that those environments need more oxygen, and it's going to start to drop off that oxygen. And as that oxygen is being dropped off, now our hemoglobin sites, let's say we have 4 possible sites, now our hemoglobin sites are going to become more and more empty because the oxygen is leaving to that area. Whereas up here, we had 100% oxygen binding. Here we have unloading. This is something that is predicted by the structure of hemoglobin itself. It's a relationship that we observe in experiments where we test the percent saturation based on the partial pressure that we have around the hemoglobin. And the important piece is that it's an S-shaped curve. It's fairly flat at the top, so we have fairly stable saturation at high oxygen levels, and then it drops off so that we have unloading in oxygen-poor environments. Pause for a second to try to draw this yourself and picture the hemoglobin saturation changes as we bind and unload oxygen. And now let's look at the textbook figure. The textbook figure, as you can see, is pointing out 2 important levels of oxygen partial pressure. So here's our PO2 of the blood. Around 40 is our PO2 found at the tissues. And around 100 is our PO2 found at the lungs. What do we do at the lungs? We bind. And we're very close to 100% saturation at that high level of oxygen. What do we do at the tissues? We drop off oxygen and we unload. And we get further down. What is the actual level at that 40 millimeters of mercury at the tissues? It's actually going to be somewhere close to about 75% saturation, meaning that we're dropping off a lot of oxygen, but not all of it. We have quite a bit in reserve. So we still have 3 out of 4 oxygen molecules bound to the hemoglobin. That means that as the tissues use more and more oxygen, we can get further down this curve and drop off more and more oxygen to them as the hemoglobin is transporting that oxygen. I hope this is helpful for you, and you can see that it's quite a range based on the needs of the tissues and the job of hemoglobin to both bind and unbind oxygen. Let's take this a little bit further. So it's not only the oxygen around the hemoglobin that can change the binding and unbinding and the saturation of oxygen. Hemoglobin saturation is also affected by other cues in the tissues that signal that more oxygen delivery will be needed. So think about how tissues can signal that they're more metabolically active. What happens when a tissue is more metabolically active? Well, it has a higher temperature. It has higher CO2. It has higher acid and higher metabolites. All of these things can actually shift the curve of hemoglobin and cause it to unload even more oxygen than it normally would just based on the oxygen levels. So here what we see in red is the normal curve that we just showed you, where hemoglobin binds and unbinds based on the partial pressures of oxygen. Now, if we add the additional signals of metabolites, acid, high CO2 levels, or higher temperature, the whole curve will shift over to the right. What does that do? Let's look at our PO2 of 40 as an example. So if we go down here for our PO2 of 40, we're back to where we were on the original graph. At this PO2 of 40, we can see that we had about 75% saturation of hemoglobin. Now when the curve is shifted by all of the things that we just listed, we can see that we actually have even more unloading of oxygen, and now we're down somewhere around 60% saturation, meaning instead of dropping off 25% of our oxygen, we've now dropped off 40% of our oxygen. That's a 15% increase in the oxygen that was delivered to the tissues because of this increase in signaling to that hemoglobin molecule. So the hemoglobin molecule is smart in a way. It senses oxygen levels and it senses other signals that the tissues need more oxygen. And when it does that, it drops off more and more oxygen to be able to fill that need in the tissues. And this curve is explaining that relationship. So overall, the effect of temperature, CO2, and metabolites is to shift the whole curve over to the right, such that the same amount of PO2 will lead to more oxygen unloading. And when we have more oxygen unloading, we have less saturation of hemoglobin, or less bound. Here's some examples of temperature and metabolite regulation. So in our first example, at normal body temperature, we showed you 38 degrees. I just showed you how high temperature shifts the curve over to the right, making more unloading for the same PO2. Low temperature can actually shift the curve to the left, because what do molecules do when they're cold? They move more slowly. So if the hemoglobin molecule gets colder in a cold environment, it actually will stay bound and not unload as much oxygen. And you can see that here in the 20 and 10 degrees Celsius curves. Obviously, those are not levels that are normal for the body, but it describes how the hemoglobin functions as a response to temperature. Here's an example of carbon dioxide levels. We showed you in the previous slide that when we increase carbon dioxide levels, showing that the tissues are more metabolically active, the curve shifts over to the right, meaning you will have more unloading of oxygen at the same level of PO2. The converse is also true. If we decrease carbon dioxide, you will have less unloading of oxygen and a higher % saturation at the same PO2 level. This carbon dioxide effect is explained in the Bohr and Haldane effects on hemoglobin. These were named for the scientists who discovered them. So the Bohr effect is when hemoglobin binds to acid or carbon dioxide, it changes its molecular structure to make it more likely to unload the oxygen. So the Bohr effect is the right shift in the hemoglobin oxygen dissociation curve that we just showed you. That means with more you get more oxygen unloading with high acid and CO2 levels. The Haldane effect discusses carbon dioxide. Hemoglobin can bind and buffer carbon dioxide and acid. So it talks more about the ability to bind the carbon dioxide rather than the oxygen. So hemoglobin will bind and buffer carbon dioxide when the O2 is unloaded. So this is actually a second part. When oxygen is unloaded at the tissues, that actually changes the structure of the hemoglobin and now allows the hemoglobin to carry more carbon dioxide. So when the oxygen is unloaded, it increases the ability of the hemoglobin to bind carbon dioxide. So the Haldane effect is about CO2 binding. So both the Bohr effect and the Haldane effect discuss the properties of hemoglobin based on oxygen and carbon dioxide. I'm also providing for you a couple of practice hemoglobin saturation curves. Practice these curves as a way to explain the binding and unbinding of oxygen and how this changes with different molecules surrounding the hemoglobin. First, with the change in oxygen, and second, with the change in CO2 and other metabolites. That's it for this lecture. Let me know if you have any questions.